Good morning, everyone. Thank you for coming by. Um, this is probably the first in a series of talks on Nobel Prizes awarded in the various disciplines. I wanted to start off discussing the Nobel Prize in Physiology or Medicine awarded to Gary Ruvkun and Victor Ambros for the year 2024. They were awarded this for the discovery of microRNA and its role in post-transcriptional DNA regulation. And I'll talk to you about the importance of this discovery, of their discovery, and how it has very broad and very important implications for biomedicine and for biology in general. Again, this is Victor Ambros on the left and Gary Ruvkun on the right. They work with Robert Horowitz's lab as postdoctoral fellows on the genetic mechanisms that underlie the development of this particular worm, right? And they have since continued their work as independent researchers in their respective institutes. Victor Ambros at the University of Massachusetts Chan School of Medicine, and Gary Ruvkun at Harvard University, right? And so essentially, their work is focused on Caenorhabditis elegans, a nematode worm, and I'll speak more about that a few slides down. And the work focused on how on the physiology and development of this worm from the embryo to the larval to the adult stage, right? And much of their work is focused has focused on the RNA regulation of physiology and development. So to sort of look at Caenorhabditis elegans, this is C. elegans gracing the cover of Science in a December 11th, 1998 issue. It's really been the model organism of choice for many genetic studies, for various reasons, right? And these are some of them, right? It's a small worm, there's a small lifespan, it yields a large number of progeny, it has a transparent body, which is very useful when you want to observe any gross changes in morphology in the body. And it becomes easier to sort of observe, make these observations and record these observations. It is also genetically tractable. Importantly, C. elegans has a very limited number of cells, right? And the fates of those cells, meaning the fates of those cells in the adult worm have been like mapped in the sense, so we know exactly which cell in the embryo is going to develop into which particular organ or tissue system in the adult, right? And so this makes it easier to follow mutations. Panels here depict the gradual progression of an embryo of the initial zygote to the initial larval stage, right? And you can see this is very well documented, and it goes from a single cell stage all the way to like an initial larval stage. And to continue that series, those transitions, here is the embryo again, and then followed by the L1, L2, L3, L4, and adult stages. Now, each of these 4 larval stages are distinct stages in the progression of the worm from the embryo to the adult. Each larval stage molds before going on to the next larval stage, and here the L4 is shown the male and the hermaphrodite versions, right? And again the male and the hermaphrodite versions. When Caenorhabditis elegans develops, or any organism develops, there is an anteroposterior axis and there is a dorsoventral axis, right? And similarly, there is also an axis along the 4th dimension, right? It's the time axis. And so when organisms are formed, when their various bodily segments and appendages are being formed, then it is signal for the formation of these appendages or organs is the gradient of a certain one or more proteins, right? But these protein gradients can also extend over time, right? And so, and it is this temporal gradient of certain proteins that drives the timely execution of the developmental program, right? And so you can have different proteins being expressed at different stages because of a certain gradient of an original protein, right? Um, and so heterochronic, I brought this term up because it implies an event happening out of turn, right? It implies an event happening at a time when it shouldn't happen. Heterochronic genes, therefore, are genes which are necessary for the timely execution of events throughout the developmental program, right? And so they therefore regulate the timing and sequence of developmental events in the different cell lineages, right? And thereby coordinate events throughout a developing organism. This is vitally important for any organism. Any disruption of this sequence can cause severe effects in the adult worm. The adult worm will be ill-formed, and so mutations in these genes therefore are a problem. They can cause cells to adopt cell fates normally associated with earlier or later times in development. In other words, what it means is that you can have the later larval stages exhibiting features of earlier larval stages, or earlier larval stages exhibiting features of their later larval stages, right? And so the first one is called retarded development, the latter one is precocious development. So again, this is just to reiterate what I just said. This is the wild type where there is L1, L2, L3 and L4 larval stages followed by the adult. This one is a Lin14 loss of function mutation. You can see that there is no L1, or in fact there is L2 where there should have been L1. In other words, it's a precocious mutant, right? The development is precocious. The cell fates or the features of L2 are exhibited when there should have been an L1. Conversely, this is what we call retarded development. So in the stage that should have been an adult, you have features of L4, right? And so this is retarded development. And so you can observe both of these in different forms. Now, typically, when you why would you come across these mutations in the first place, right? Typically, in a study that is using a genetic analysis, the approach is to create a large number of mutants and screen for mutants, and then look to see how they differ from the wild type. Now, you can induce mutations through a chemical, which is a chemical mutagen, or through radiation, which is an ionizing radiation, and then look to see what changes in morphology or physiology happen in comparison with the wild type as a reference. Right? And so these were the mutations that were observed with respect to the wild type. This is the wild type here. This is one mutant that they observed in the genetic screen, right? The Lin4 mutant. The Lin4 mutants, they will exhibit features of what we call retarded development. So features of L1 will show up in L2 or L3, right? Whereas this is Lin14, right? These are small mutants. They will exhibit such a morphology as this, and they lack larval development because the Caenorhabditis elegans is a transparent organism. You can observe these changes. This is a reporter construct essentially, and the quantity of protein is directly proportional to the intensity of the blue light that you see here. Right? And so you see here in the regular wild type, like here and here, there's an X-gal, which is a dye. You see it staining on both sides, right? When you look at the Lin14, this is a wild type Lin14, you can see that it's expressed in the first L1 stage, but not in the later larval stage, right? However, when you have a mutant, when you have lin14, where lin4 is mutated, you can observe that there is an expression of lin4 where there shouldn't have been, right? The last panel suggests that you're having an expression of protein, of lin14 protein, when it should not. Ideally, this should be what happens and not this. What this will cause is the retarded development. Essentially, the worm will exhibit features of L1 in L4. Okay, and so after much work, on these, it was observed that these 4 heterochronic genes, lin4, lin14, lin28, and lin29, are necessary for the post-embryonic developmental events in C. elegans. Their normal activity is important for these developmental events to unfold at the right time, right? So, I mean, the next question was, especially in the case of the lin4 and lin14 mutants, do they give rise to the same phenotype, right? Turns out it doesn't. lin4 is actually a negative regulator of lin14, meaning when lin4 levels are high, lin14 levels will be low. If you overexpress lin4, lin14 levels will go down. And this is often done using what we call epistatic analysis, especially when a single outcome is controlled by more than one gene, right? And when you compare the result of multiply mutated genes with those of singly mutated genes, is there a difference? Does the effect of one gene come before the other? Are they interdependent? You can analyze these and parse these effects using epistatic analysis. And when they do that, they see that it's a negative regulator of the lin14 protein, right? Meaning when one goes up, the other goes down. So how exactly is this happening? I mean, is there a lin4 protein in the first place? What kind of regulation is it exerting on lin14, right? And so to determine the nature of the lin4 effect on lin14, they first went about analyzing lin4 itself. So you can see here that lin4, this is the northern blot analysis showing the lin4 RNA exists in 2 different forms, right? There is a longer form that is about 61 nucleotides long that is then, processed in the cell to give rise to a 22 nucleotide lin4 transcript, right? And that is the active form. That is the form that is predominant in the cell, right? Did this RNA code for a protein? It did not, right? I mean, the lin4 RNA does not code for a protein. It does not have an open reading frame. There is no protein. There is no evidence of protein in the lin4 in the cell, essentially. And you can rescue lin4 deletion mutants. So if that gene is ablated, then you can rescue it with just lin4 RNA. Okay, then all these elements of evidence pointed towards an RNA-based role for lin4. Right? And so question remained, how exactly does lin4 regulate its effect, the expression of lin14? Now, we didn't know at that stage about how it was affected, right? Typically, transcriptional regulation or regulation of expression of a certain gene occurs at different levels. You can have regulation at the transcriptional level. Actually, you can have something before that as well. You can have regulation at the transcriptional level, which is normally known to be affected by transcription factors, which are proteins. Then you can have regulation exerted at the post-transcriptional level, where the mRNA, the lifespan of the mRNA is regulated, right, by some other protein or molecule. You can have translational regulation and then again post-translational regulation, right, all along this central dogma. So transcription regulation would happen here, post-transcriptional at the level of the RNA, translational at the level of protein, and then post-translational at the level of the synthesized protein, right? And so it's worthwhile looking at the structure of the mRNA at this point. So the mRNA has a 5' end and a 3' end. If it's a coding protein, correspond to the starting and ending regions of the protein. At the 5' end, there is a cap, and then on the two ends of the open reading frame, which is actually the portion of the gene that codes for the protein, there are two regions called the untranslated regions, shown here in green. Right? One is the 5' UTR or untranslated region, and the other one is the 3' UTR. The 3' UTR is known, followed by a poly A tail. So 3' UTR is known to be involved in the post-transcriptional regulation of mRNA, and the direction of translation is from the 5' end to the 3' end. Right? If students will recall that the 5' end actually corresponds to the N terminus of the protein, and the 3' end to the C terminus. Right? And so at this stage, fortuitously for us and for science, Gary Ruvkun and Victor Ambros had a phone call, and they compared the sequences of the Lin4 RNA and the Lin14 mRNA, and they found regions of complementarity at the 3' in the 3' UTR region of the Lin14 mRNA, right? And this was the first indication that the effect that Lin4 has on Lin14 might actually be exerted by physical contact between the two forms of RNA, right? But this complementarity is not absolute, meaning there's not perfect, sense, antisense, or perfect complementarity between these two regions, right? There are regions which are not complementary and which form these bulges, right? So again, this is the schematic of that same. At that stage, it was still a hypothesis. They weren't sure, but subsequently, and this is again a more detailed picture showing the exact sites of the Lin4 binding, right? But I get ahead of myself here. To sort of step back, there are different forms of analysis that indicated that this was really the way the interaction was happening. This was really the way Lin4 was exerting its effect on Lin14. One was what we call a genetic analysis. So if we delete the Lin4 gene, there's no Lin14 levels. They don't go down from going from L1 to L4, right? There were other pieces of evidence as well, and I've obviously gone over this before, but to come to this, right? So when Lin4 complementary sites on the Lin14 3' UTR of the mRNA are deleted, right? Then there's no more repression, right? Confirming that Lin4 binding to Lin14 mRNA happens through the UTR, right? And so essentially, so just to say that again, if you come to this, when you delete Lin4 complementary sites from the Lin14 mRNA or Lin14 mRNA 3' UTR, then there is no more repression of Lin14 levels, indicating that Lin4 repression acts via the 3' UTR of the Lin14 mRNA. Okay. The researchers went further to do a reporter assay where they fused the Lin14 3' UTR to a reporter construct, in this case XGAL, and check to see how it functions in cells which have wild type Lin4 and mutant Lin4. So in wild type Lin4, there is a down regulation of the dye of the reporter construct, right? Reporter expression is down regulated, but in Lin4 mutants, Lin14 expression persists, right? Indicating again that the 3' UTR is a very important component of this repression, indicating especially post-transcriptional repression via the 3' UTR, right? Northern blot analysis also indicated that the Lin4 microRNA was expressed at the same developmental stage, at the developmental stage when Lin14 was repressed, right? Or when Lin14 protein levels were decreased. So indicating that there was a concomitant, there's a correspondence there indicating that one acts on the other. Right? Immunoprecipitation. The Lin4 microRNA appears to be pulled down with the Lin14 mRNA along with proteins from the RNA-induced silencing complex that accompanies these microRNA. Right? So all that put together, the authors to propose some things, and these were like very novel findings, that the post-transcriptional regulation of the heterochronic gene Lin14 by Lin4 mediates temporal pattern formation in C. elegans, and that the C. elegans heterochronic gene Lin4 encodes small RNAs with antisense complementarity to Lin14, right? And so these were really path-breaking studies at that time, and which eventually led to their being awarded the Nobel Prize. So again, to put all of that together, as Lin4 levels rise, Lin14 levels drop. And subsequent to the identification of Lin4 microRNA, a second microRNA was discovered in C. elegans. This is called LET7, right? And again, you can see how the expression of LET7 causes the decrease in Lin14 protein levels, going from the L3 to the adult, right? They both work similarly in the execution of the developmental program, right? And so this was cloned by Gary Ruvkun's lab, and this is just a picture showing how the LET7 happens to be across a large number of animal phyla all the way to the Homo sapiens, right? Now this was actually even more novel. I mean, for a long time, the authors knew that Lin4. I mean, they wondered whether Lin4 was something very niche, something that happens only in C. elegans, and whether or not it had any direct bearing on other animals. You know, was it just a one-off, right? And so it turned out with LET7, the discovery of LET7, and with the phylogenetic analysis, that indeed it is actually a fundamental form of post-transcriptional regulation in other phyla as well. And this is again to reiterate what I said, there's an evolutionary conservation of the LET7 expression and microRNAs more generally, right? That is all well and good, but how is the discovery important, especially to biomedicine? It's important, first of all, because it opens up an entirely new realm of gene expression regulation, right? Or gene regulation. Secondly, there are biomedical implications for these discoveries, right? They are now known to be potential disease markers for various diseases, because there's a very disease specific expression of these microRNA in the circulation and in tissues, right? MicroRNA are also involved in tumors as tumor suppressors or oncogenes, and can be targets for different drugs, right? They are important, they influence the growth and repair of different tissues, and in such cases they might be important for the management of cardiovascular disease, right? Dysregulation of microRNA levels have also been linked to various neuromuscular diseases or neurodegenerative diseases such as Alzheimer's and Parkinson's, and they could possibly, provide us a handle for potential interventions. MicroRNA profiles and the stratification of patients, can we use it appears that we can use microRNA for stratifying risk for individuals with various diseases, especially in the area of precision personalized health. So I think that I'll wind up the discussion with that slide, and I'll be happy to take any questions you have. Thank you all again for coming by, and I wish you a good afternoon. Thank you all.